All right, thank you all so much for the invitation to talk here. At least I've been to Michigan for about a year now. At least 4 different people have said you need to meet Tim. And I never did, and now I get to meet Tim. So if this was about bringing people together, well, at least for Tim and I, that worked out wonderfully. I'm going to talk about some things we've learned from doing closed-loop AI-driven experiments in our lab. So we've done about 1 million experiments now on microbes in the lab using robots and AI generation. And I want to turn that into some things advice I would give to my former self if we're doing this again. And right now the system is down while we re-engineer the software based on lessons we've learned from the first 1 million. So if I was trying to get you to click on something on the internet, I would say that, there's this one thing you can do to improve our performance, and it's not regularization or bootstrapping or gradient boosting or anything like that. The answer is you just need better data. And I think this is overlooked a lot because in part, most people say, well, we're awash in data to begin with. You know, who's thinking about we need more data in the age of big data? And the second, I'm guilty of this as an educator, that when we train people on machine learning, we give them the data, or we say, go find a data set. And it's not an option to say, well, maybe I need to get other data, or I should collect new data or run more experiments. So these are the 3 lessons I'm going to talk about today. I'll go through them one at a time. And we're sort of taking a split now. I like that the dichotomy was brought up before between found data and then design data. This is on the design data thing. We have control over our experiments, and it turns out that as scientists, we often make very poor choices when we're collecting data. So I really like AI history, and I read about a lot of this over the last couple years, and there's been 2 AI winters, which are things where we promised that AI was going to do amazing things, and then it crashes. They're kind of AI recessions. The funding dried up, people left the field, other people went into hiding to continue the important work of AI, and then reemerged. So I was thinking about, well, what's going to cause the third one? Because I do this for 2 reasons. One, if there isn't a third AI winter, we're all going to be so busy that no one's going to remember that I said this. But if there is a third AI winter, you'll say, oh, that guy in Michigan predicted that this was coming. So I kind of win either way. And I don't think in between the first 2 AI winters, they were thinking, I wonder when the second one is going to come. So what's going to cause the AI winter? And I think it's something called a data drought, which is a made-up term, so I'll explain what it is. So a data drought is like an actual drought, and I apologize if anyone is here from climate and space sciences that knows more about droughts than I do. It's a local shortage of data. So we have lots of data, but there's going to be areas, including the field that I work in, where we have a shortage of data. Just like there was a drought in the southwest United States, but it still rained in Michigan. It's going to appear. I work in microbiology, particularly in the microbiome, and especially the oral microbiome. And we're suffering an enormous data drought. We don't know anything about a lot of these microbes. There are 46,000 species of bacteria that we've identified, and our group did a big bibliographic analysis, and we pulled out every paper that's been written on the microbes, and we counted how many papers are for each one of the microbes. Well, 50%, half of every microbiology paper that's ever been published is on 13 species of bacteria, 13 out of 46,000. And 3/4 of the species of bacteria that we know of have never been a subject of a scientific paper. So if you hook this up to a language model and you throw all of PubMed into a language model and you ask about a bacterium, 3/4 of the time, there's nothing for it to read. There's nothing for it to learn from. I say, well, that's all right. This is going to fix itself because everyone's drowning in the literature right now, and the number of papers is increasing almost faster than inflation. But if you normalize that to the rate of discovery of new species, right, we're discovering so many new species of bacteria that the knowledge density, the number of papers published per species, has decreased by almost 1/3 over the past decade. So we're in an ignorance explosion. We know less and less about more and more different bacteria. So that's the first part, is it's local. It affects me. I'm selfishly talking about my field. Other areas that this might not happen at all. The second thing is that there's a balance between supply and demand in water. It could either rain less or you could use more for agriculture, and then you run out of water. And we have the same idea, is that there's a balance between the production of data and the consumption of data. There was this great paper put out by Google. It's a qualitative research. It's all done with interviews of data scientists from around the world. And what they found is that everyone wants to work on the models and no one wants to work on the data. And they identified lots of reasons. There's career issues. You're not going to get promoted by curating data. You're going to get promoted by coming up with a new machine learning technique or new neural network technique. There's all sorts of issues with collecting the data. And they see even in very important areas, we're all using the same data sets over and over again. So although water, only one person may be able to use it, I think data is the same way. We can't just all be using the same data or we're all going to be biased. This is data collected from our lab. Each one of these triangles is a plot of what happens when you take away a pair of amino acids from a bacteria and then try and grow it. All right, so there's 20 proteinogenic amino acids. You take 2 of them away. Along the diagonal is just if you take the single amino acid out. These are 4 different species of bacteria. This is done aerobically and anaerobically is the top and the bottom. And all you should notice here is that there are different patterns of auxotrophies, meaning they require different things. The dark boxes, they don't grow. The light boxes, they do. So for each one of these species to say what amino acids does it need to grow, that depends on the different species, okay? Now the interesting thing is that we built models for all of these bacteria. We use the Department of Energy knowledge-based system that Ian referenced. We build these metabolic reconstructions. We manually curated them. It takes a year for the first one and 4 months for everyone to make sure we got all the reactions right. The models all say these 4 bacteria are identical when it comes to amino acid auxotrophies. They predict that the boxes are all white for them. They have complete pathways to make every amino acid. There's no reason they should have to take it up from the environment, but for whatever reason, they choose not to. They have regulatory systems that are now reflected in the model, and we can't predict any of those auxotrophies. We didn't know they existed until we measured them in the lab. And you might say, well, that's all right because we can reuse data from other similar species. Well, these are as similar as you get. These are all, the S is for Streptococcus. These are all closely related bacteria. They all live only in the same spot. They live on the surface of your teeth. They have throughout their entire evolutionary history. They can't live outside of your mouth. Right? So they live in the same place, they eat the same thing, they are very closely related, and they're entirely different, and I don't know of a way to predict it other than actually doing the experiment. Right? So reusing the data isn't an option for us here. So I think that's what's going to happen, is we need to concern ourselves that in certain pockets of science, there is no big data, there is no super influx of data. We need to talk about a generation. Okay? The second lesson that we learned is that if we have to generate huge amounts of data to solve this drought, we have to do this sequentially. We have to do this adaptively. We can't just fire up a massive screening campaign and say, let's do all the amino acid screens. The triangles I showed you before, each one of those triangles is 213,000 liquid handling operations to generate the data. We say, well, let's just do it 1 billion experiments and collect all the data. That doesn't work. How many people have done one of these before, played a Wordle? It seems like quite an impossible task. And I said, all right, it's a 5-letter word. All right, so if I picked someone out of the audience and said, I want you to write down 5 guesses, or 4 guesses only, 4 5-letter words. I'm thinking of a 5-letter word, and then you hand me a slip of paper and I'll see if it's on there. And you say, that's ridiculous, that's not going to work at all. All right, but Wordles are in fact solved. There's a deterministic algorithm, of course someone figured this out, that in a Wordle, if you get the feedback from the first answer, you can adapt the second one, and you can figure out any 5-letter word that was used in the Wordle database by following a decision tree diagram. A group of computer scientists figured it out. And the reason is you don't have to guess all 4 at the start. You can guess once and then choose how you refine your second experiment. Something that goes from really impossible to now, it's solved, it's deterministic. So we use this in the lab for our self-driving robots. We ask biological questions, and then we allow the robots to do a sequence of experiments to answer the questions. And we are inspired to do this by the advances in self-play that underlie a lot of the AIs that have won all the big board games. So this is one that talks about when they beat the world championship in Go. And they use a technique, an AI technique called reinforcement learning, where you make a guess, you make a move on the board, and then you get a signal, either reward, that was good, you won the game, or no, you lost the game, that's bad. And then it has to learn to say, well, I'm not going to do that move again because I lost the game. And this goes in a loop. So we do the same thing in the lab. The question is, what does self-play mean for science? So DeepMind that did this, they pointed two of the agents together and they say play against each other, and over time they learn to get better and better. We can't do that all in simulation, so we need to have a physical manifestation. And the second is this trains agents that know answers, and human, we want to know the answers. We don't want agents that know science, we want the answers. We have to solve that as well. So our solution is we have an AI agent that thinks it's playing a game. It makes moves. It's trying to maximize some sort of score, but is really controlling robots in the lab. So it makes a move and says, I want to do this thing, and it's really requesting an experiment. The experiment gets run by the robots and the data comes back. It learns and it requests new experiments to try and maximize a score, and it runs in a closed loop. We have about 14 different robots that we use to run the experiments that are hidden behind the wall that the AI doesn't know that exists. If you've ever read the book Ender's Game, I won't reveal the ending of that, but it's the same idea. It's very different on the other side. We started this up and we said, well, we can do about 300 experiments a day, and then by upgrading the robots now, we're up where we can run 10,000 experiments a day, all at the request of the AI system. The hard part of this is that it has to be able to pick any experiment it wants. Reinforcement learning only works unless you let it make mistakes. So we can't say, oh, we'll just do 10,000 experiments, here's the data. We have to wait until it says, I want to run this experiment, and then we have to execute it right away. Every day at 9 a.m., a technician comes in, collects the data from yesterday. We put the data back into BacterAI is the name of our system. It can think for a while and it gets 1 hour to plan. Because by 11 o'clock we have to start the scheduling and the robots to dispense so that they can inoculate the cultures. All these experiments have to run overnight. We have to give time for the bacteria to grow. So there's a lot of engineering that went into getting the times on this down where we can run in a closed loop every day. Adam Dama was an undergraduate in the lab who stayed for a post-bac, which is the time after graduating with his bachelor's degree, for 2 years, and he was the main architect of the robotic system. So here, we're going to watch BacterAI learn. So this is, every circle on here is an experiment. It's showing the number of amino acids that it took out of the media. Here we asked it to learn what amino acids a particular bacterium needs to know. The filled circles means the agent predicts that it will grow, and the open circles predicts that it will not grow. If it was a black circle, it was correct once the data came in. Red circle says it was incorrect. So by day one, it guesses purely randomly. We don't know anything about the amino acids, so we just set up a random neural network and let it make random guesses and flail around and know that it's going to get the answer wrong. Just like when the chess playing agents start, they make random moves. They don't have any knowledge of how the game works. So about half are wrong. This is now on day 3, and you see already it's getting better. It's changed the distribution. It's now picking fewer experiments that it gets wrong. We don't allow the robot to repeat experiments. So the easiest way for it to score points would be after it's seen the answer to request experiment. It can't do that. If it picks one it's already run, we say guess again, choose a different experiment. And then this is, we'll watch it go every other day here. We're showing the results. It's not monotonic. It will learn something and then it looks like it's getting worse. The predictive accuracy goes down. I would liken this to maybe a chess player that learns a new opening and tries it in a game and loses the game because they aren't quite experienced with it yet. We see the same thing in our robot. By day 13, it's 90% accurate. So 90% of the time it can tell what the experimental result will be before it requests the experiment, and then we stop the agent and say, all right, that's good enough. Inside the agent is a neural network, a very simple feed-forward multi-layer perceptron that predicts the result of the experiment, not if it will grow or not, but actually how much the bacterium will grow. So this is a plot of the neural network. Each dot here is a single experiment. The blue line is the experimental prediction, the black line is the experimental result. So at day 1, these are random predictions. The network has not been trained because we don't have any data. So it predicts just a random growth level, and there's no correlation between the blue dots and the black dots. So with perfect accuracy, the black dots would be right on top of the blue dots. And then you see, as it goes along, this is the same time course I showed you before, but now we're watching the network on the inside. By day 13, it has a network that's very accurate. It can tell you exactly how much the bacterium is going to grow. Okay, so now it knows. The agent can tell us exactly if a bacterium is going to grow. We want to find out as well. So we run a second AI agent whose job is to pull the information out of the first one and construct for us a human understandable rule that we can use to learn what the growth requirements are of the bacteria. So this is one example. This is a bacterium lives in your mouth. It's a good bacteria. We actually want this one around. And this is a simple logical rule that represents the knowledge in the AI. It's not as accurate as the neural network because we've regularized it. We said you have to make it simple enough for a human, but this is the rule. If you restart it, we ran it on a second bacterium, you get a different rule again. And this was not surprising because we knew they had different oxytrophies after that first set of data I showed you, but we don't know why 2 bacteria in the same place, in the same environment, have developed different oxytrophies when they have the capability to have no oxytrophies. They have all the genes in their genome. And so I want to impress on the second lesson here that doing experiments in a series is much more powerful than just a high throughput screening campaign. We've calculated by experiments that we're about 1,000 to 10,000 times more powerful by doing a series, meaning we can simulate from the data. It takes us up to 10,000 times more experiments to learn if we collect them all at once at the beginning, to learn the same amount of information than if we let the agent get better and learn from its mistakes and plan better experiments every time. Okay. The last lesson I would make is that it's said in a joking way that experimental data is too important to leave up to the experimentalists. That's said with about 2/3 of our lab is experimental, because the way you should design your experiments is not the way that science teaches you, because you should change everything, all things, all the time, all at once. This is because there's this law of bounded estimability, which is not a real law. I don't know the name of this law, and if anyone knows the actual, like a law that says the same thing that's true and knows the name, I would love to know it, because I'm teaching a class and we refer to this. That if you're building a computational model, the degree of the interactions that you can estimate is always bounded by the number of things you have moved simultaneously in your data, if you're building the model straight from the data and only from the data. So I had a collaborator who came into my office one time and said, I did this experiment where I mutated every single base in a protein, and then I measured how well it bound and cut some DNA. So I did a whole bunch of experiments. I changed every single position one at a time, and now I want to predict what combinations I should do in the final protein to give the highest value. So can you do some machine learning on that? And I said, well, no, absolutely not. There's no information in your data about the interactions. I couldn't predict anything about the interactions. And he said, but no, that's what the machine learning is for, is to get the interactions. And I had to say, well, unless you have some biophysical model we can couple with it, there's no information about interactions because you did one at a time. But that was a good scientific thing to do. I like to joke that, would a biologist be able to train a language model? This is a parody of a great paper that was written about, can a biologist fix a radio? For language models, we always want to predict the next word or the next token, but you can think about it as a word. So if a biologist was generating the training data, they would say, all right, I took this sentence, it's a very important sentence, this is a model sentence that we can use to understand human language, and then I took one place and I cycled through every word, and I did a high throughput fluorescent reporter assay to determine if that word made sense in the sentence. And then using these data, I can now predict the next word, and we'll train a language model off this. Of course, this wouldn't work. Right? Language models require just rich corpus of data in order to predict. And they would present this and someone would say, did you do my sentence? Because I have a favorite sentence that I'd like you to do experiments on. Because you can't just change one thing at a time. It's easier to learn if you change all the things at once. The benefit of changing all the things at once and making our design data look more like found data, where we have no control and patients come in and have all their covariates and we have no control. The advantage is that you get excellent scaling in terms of number of factors. So we were worried with our system that we started by studying amino acids, and there's 20. There's 1 million possible combinations of amino acids. The growth medium actually has 39 different ingredients in it, sugars and salts and minerals and vitamins and all the other things the bacteria need to grow. So we thought to go from 20 to study the metabolism of 39 different ingredients, that's an increase in the solution space of 500,000. And we were worried it was going to be an explosion in terms of how much data we need to collect in order to make sense of it all. Well, it turns out that to double the number of inputs and learn the response, with a little help from transfer learning, it only takes twice as much time. The solution space expands combinatorial, but the amount to learn it only increases linearly with the number of factors, because all of our data are multifactorial. It's changing all of the factors all at once. And this isn't always guaranteed, but I think it happens because properly designed experiments are multifactorial rather than if we'd gone in every single one at a time. So to wrap up, there's lots of corners of science, and especially in biology, in my corner especially, where there is just no data from which to train the models, and we have to think about how we generate large compendia of data. If you have the right data, the learning is relatively easy. We use very simple machine learning models to make predictions from our data sets. And when you're thinking about designing data, if you ask a high throughput screening person, they'll say, well, we want to reduce the cost of the assay so we can maximize the number of experiments. And I said, we need to think about the information content because well-designed, planned, sequential experiments are worth much more. They punch above their weight than just a high throughput screening campaign. So with that, I want to thank everyone in the lab who worked on building the projects, and of course the funding sources and all the organizers for putting on this great symposium. So, thanks.
So we do have time for a couple of questions.
I love your slide where you had Jensen showing in the various fields of the microbiology. So I see there's a lot of opportunity there for, if you're trying to grow into a new field. But my question was, and I agree, there's a lack of data, but is there a way to leverage the data that may be a prediction? Could that bring some of your modeling close to the real life? Is that an option, or?
Yeah, so we started by building the models and trying to make the predictions, and then we originally went in to do the validation, and we found that we're just way off. I think there is still a lot of value in biochemical databases and models and everything to avoid us relearning things that we already know. Here we're starting completely from scratch, and we have to piece everything together on their own. It would be good to say, the model says this, my data seem to agree with the model, we should stop spending experiments over there because that's inconsistent, and then focus on the surprising things that are unpredictable. So I view sort of models now as kind of a prior, like what we can subtract out before we start running the experiments after.
I'm in the sequencing space, so I don't know, I would probably ask you if you've explored the sequence read archive. I didn't see if it was listed in your data sources at the end, but I wonder if, because it's very challenging to even find some of these microbes, right? So it's very hard to even design an experiment to go detect them, right? So there still is a lot of unknown that we don't know, and I understand that's scary. But I would probably also see if we can look into some of the new sequencing data that is coming out of some of the Illumina and the PacBio to see if it has any more data for your modeling.
Right. I agree. From primary sequence, from genome sequence, we don't seem to be able to predict it. We have a student now who's trying to do the same thing using transcriptomics. So where the experiment is a transcriptome, and then can we learn to predict the gene expression That's a harder challenge because we can't do 10,000 of those experiments every day. We can do, you know, a couple hundred of those a week, but the numbers are small, and we have to be much better about planning what's the most informative next experiment to do. So yeah, I agree with you, absolutely.
Yeah, mind-blowing stuff, really, really cool stuff, Paul. One's a simple technical question. Everything seems to be well-plate based, but you were talking about maybe one experiment at a time. Do you run singletons, or is everything, like one experiment is 96 or 3, 4?
We do, so because every experiment takes 24 hours, it's allowed a batch size. So usually between 336 to about 1,000 experiments per day,
Right.
it gets to pick. And that actually makes the experimental selection a lot harder. Saying the next best experiment is easier than saying what's the next 1,000 best experiments. They're all running 3D4 well plates.
Then another question on the one slide you showed with the oxytrophic relations, where it was matrices they were, you know, the model before you came around was predicting everything would be white, and then you showed that there's a couple dependencies. Can you reason that now, having seen that, or does that mean kind of teach about the biology of what's going on? I mean, that's a big question, I guess, but can you see, like, even though this machinery is in that bacteria, it's not active, or like why did the models get it wrong before?
Right. The way we're going about synthesizing all of that is we're training neural networks that learn to fix the model. So we take the model and then we say, you have access to all this data, these are the results, you make a prediction, we'll tell you if it's right or wrong. And we're doing the same reinforcement learning approach to say, what changes would you have to make, what would you have to add, what would you have to take away, until the model now recapitulates the experimental data. That's hard with, it's easy to overfit the model to get it to work, but we're less worried about that because you can keep always asking more data. You can always say, we'll make new predictions, we'll run it, and then train on that, rather than fitting it all to one data set. So that's our solution, because it's kind of, there's about 100 or so enzymes that are involved in amino acid biosynthesis. So the answer is somewhere in the 100. We think it's 8. We've got it down to about 8 enzymes that seem to explain the majority of the effects.
All right, let's take a